Hello students, I am Amruta Pimpalwadkar working as an assistant professor in the Department of Biotechnology, KTHM College, Nasik. Today we are going to discuss a practical from the course Practicals in Food Biotechnology, which is designed under the UGC stride scheme. And the topic of our practical is aflatoxin determination from spoiled groundnuts. So why we have this topic in our practical course? So in theory, we have discussed that microorganisms are present everywhere in the environment. So similarly, they are present in the food also that we consume on the daily basis. So if these organisms multiply in our food, then they will result into the food contamination, and this food contamination will result into the food spoilage, and this spoiled food we cannot consume. So in this practical, we are going to discuss first what is aflatoxin and how it has role in the food contamination. Then we will discuss how it is extracted and how we can detect qualitatively that aflatoxin is present in the given food sample. The food sample that we are going to take is spoiled groundnuts. Then we will see how we can determine its concentration present in the food sample. That means we are going to perform quantitative detection by competitive ELISA technique. So what are the learning objectives behind this practical? Firstly, you need to know the concept of mycotoxins. That is, they are the toxins which are secreted by the fungus. Then we are going to see what are their structures and what are different types of these aflatoxins. Then we will see the extraction method for extracting this aflatoxin from the spoiled groundnut. Then we will first see what is the principle of ELISA technique, and one type of ELISA technique is used for determining concentration of aflatoxin from food, that is competitive ELISA. So we will discuss the application of this competitive ELISA for determining quantity of aflatoxin present in the given food sample. So to start with, what are aflatoxins? So, before going to aflatoxin directly, we will discuss the concept of mycotoxin. Now, if we see, there are two parts of this word. One is myco, that refers to the fungal origin, and second is toxin, that means the substance that has hazardous effects if it is consumed by human beings. So, these mycotoxins are the secondary metabolites of the fungus. Now, there are two types of metabolites basically. One is primary metabolite and second one is secondary metabolite. Primary metabolites means these metabolites are required for survival and proper functioning of the cell, and secondary metabolites means these are not required for survival of the cell, but if these metabolites are produced by the cell, then it is going to give certain added advantage in their living. So example of this includes antibiotics and toxins. So similarly, these mycotoxins are produced as a secondary metabolites by different fungus. So major fungal genera that produce this type of mycotoxins include Aspergillus, Fusarium, and Penicillium. So why these are basically called as toxins? Because they have adverse effects on the human beings if they are consumed through our food. So basically they are causing food contamination, and if we eat such food which is contaminated with this fungus, then we are at the risk of consuming these mycotoxins along with it. And what are the effects caused by these mycotoxins? That is, they are carcinogens. That means they are cancer causing, and basically they affect our liver. So they are called as toxins. So one type of mycotoxin is aflatoxin, which is mainly found in cereals, corn, peanuts, cottonseed, and nuts. So, which are the fungal types which produce this aflatoxin? They are Aspergillus flavus and Aspergillus parasiticus. So, the picture shows you the growth of Aspergillus flavus. Left one is its growth on the agar plate, and the right one is the weight amount of this fungus, how it looks under the microscope. Now, these aflatoxins chemically are the coumarin derivatives. What is the meaning of derivative? That means this basic structure will remain same, but there will be certain functional groups that will be added. So, based on the fluorescence, there are two types of the aflatoxin molecules. That means based on the color of fluorescence. Now, what is the property of fluorescence? These aflatoxins can absorb the light of a shorter wavelength, and they can emit the absorbed energy in the form of visible light, that is light radiation having higher wavelength. So there are two colors which are emitted, one is blue and second one is green. So B type refers to blue color fluorescence and G type refers to green colored fluorescence. So on screen, you can see the structure of these two types, that is B type and G type. Now these two basically are subdivided into two more types based on their structural differences like this. Then one more type that you can see on screen is aflatoxin M1 and M2. This is basically the hydroxyl derivative of the B type only. So there are basic two types, B type and G type, and M type is the derivative of B type. Now, any hazardous compound has regulations because see, this aflatoxin will be present in the food, and if you are consuming it in higher concentration, then what will happen? You will have some diseases or disorders which are linked to this aflatoxin. So there are some regulations to their concentration which is present in the food. So concentration of aflatoxin that is permitted, that is maximum tolerable limit of aflatoxin in food is zero point 0.5 micrograms per liter or zero point 0.5 parts per billion, that is ppb. Now this aflatoxin, to know how much is the quantity of aflatoxin present in the food, we should detect it first and then we should be able to determine its concentration also. So there are various methods available like given on the screen. One is fluorescence detection, that is for just detecting the presence of aflatoxin. It is not quantitative method. And then quantitative methods are thin layer chromatography, high performance liquid chromatography, gas chromatography, and enzyme linked immunosorbent assay, that is ELISA. We are going to discuss ELISA for quantitative determination of aflatoxin in food. So to start with, our practical has two parts. When we take spoiled groundnuts or the food sample that is supposed to contain aflatoxin in it, first thing that we need to do is we need to separate it from that food component. So first is the extraction of aflatoxin from food sample. So how you will extract this aflatoxin now? We are going to extract it by using suitable solvent in which aflatoxin will be selectively soluble. What are the requirements for this? First thing for extraction that you need is infected material, that means spoiled food sample, that is groundnut. Now it is finely ground for better extraction because if particle size is smaller, then it gives higher surface area, so better extraction is achieved. Then second requirement is methanol is to water mixture in the proportion 55 to 45. Now methanol is the organic solvent that can be used for dissolving aflatoxin present in the food sample. Then next is Wattman filter paper number 1 for filtration. Then chloroform again acts as an organic solvent, and N-hexane is added because it removes fats which are present in this mixture. What is the protocol now? First thing that you need to do is you need to take groundnuts, spoiled groundnuts, and remove their outer coverings. Why you need to remove their outer coverings? Because it is a fungus, so for protection it resides under the coverings of the groundnuts, so you will get good growth under those coverings only. So first remove those coverings and crush those groundnuts into paste. Then second thing is you need to soak this into the mixture of methanol and water and give at least 15 to 20 minutes so that enough time is given for the extraction of aflatoxin into the mixture, and then you need to filter that to remove the particles present. After that, take the filtrate. Now in that filtrate, you need to add 1 ml of hexane, that is going to remove fats present in that. And second thing that you will add is 1 ml chloroform, because we have seen that it is soluble in organic solvent. So chloroform will act as a solvent here in which aflatoxin will be miscible. Then shake that mixture vigorously and allow it to stand for at least some time so that two layers of hexane and chloroform will get separated. Now after separation into two layers, you are going to take the drop of upper layer which is organic, that is of chloroform on the TLC sheet. TLC here will TLC sheet, which has silica coated on it, will act as a adsorbent here. And second thing that you need to do is, as given in the picture, at T, that is test, you are going to put a drop of your mixture extracted aflatoxin, and as a control you are going to put the drop of hexane and chloroform mixture alone. Now this will act as a control, because we need to compare this with control to see whether there is fluorescence exhibited with these solvents only, so that to avoid false interpretation. And then you need to observe this TLC shade that contains both the drops, control as well as test, under the UV light to see whether they fluoresce or not. So if you have got aflatoxin in the chloroform, that is, if it is extracted properly, then it should give fluorescence like it is given in this picture. So if you have extracted B type of aflatoxin, then you will get blue colored fluorescence. So how you are going to interpret your observation? You will interpret as, if no color is observed, that is no fluorescence is observed, then you will say that no blue color fluorescence was observed in the region of control on TLC shade. And now test circle will indicate actually whether your aflatoxin is present or not. So if fluorescence is seen, then the blue color fluorescence can be observed in the test region, and if it is not seen, then blue color fluorescence cannot be observed in the test region. This is just the observation. Now based on this, you need to interpret your result. How you will interpret your result? Now if fluorescence is present, that indicates that aflatoxin is present in the given food sample and it is extracted properly. If fluorescence is not present, that means in both the circles you are not getting any kind of fluorescence, then that means that aflatoxin is not present in the sample, or it is not properly extracted. So you will interpret your result as, the blue color fluorescence in the test region indicated the presence of aflatoxin in the extract prepared using given spoiled groundnuts. That means your extraction procedure is successful. And second thing is, the absence of blue colored fluorescence in the test region indicated that the aflatoxin was not extracted. You can give negative interpretation also, because extraction of aflatoxin is not that easy. You need to take highly spoiled groundnut sample for extracting this in greater amount or greater concentration. So this is first part. What we have done in this is, we have extracted aflatoxin and we have done qualitative detection by observing its fluorescence under the UV light. Now next part is, okay, we have got the presence of aflatoxin in the food sample. Now next step is to measure its concentration. So next thing is, you need to do quantitative determination of aflatoxin. So this is how you are supposed to proceed in stepwise manner. So for quantitative determination, commonly ELISA technique is used. We have seen that the modification of ELISA that we are going to perform is competitive ELISA. But before going to that competitive ELISA technique, first you need to know the basic of ELISA technique. So first we will see the principle of this ELISA technique. Now what you need in this ELISA is, basically the full form of ELISA is enzyme linked immunosorbent assay. Now enzyme linked, so enzyme will be present, so specificity of enzyme will be utilized, and this enzyme is such that it acts on chromogenic substrate. Now what is chromogenic substrate? That means the substrate, when it is converted into product, it will generate color. So such type of substrate is called as chromogenic substrate. Here the enzyme that we are going to use, or commonly used enzyme in ELISA, is HRP, that is horseradish peroxidase, and its substrate, its specific substrate is TMB, which has a full form 3,3-5,5-tetramethylbenzidine. When this TMB is converted into product, it gives blue color. And third thing that you need is antigen coated microtiter plate. Now name indicates that here immuno, that means you are going to use the specificity of antibodies, and enzyme refers to the specificity of substrate and enzyme. So how you are going to proceed now? First what you will need is, you will need a microtiter plate on which antigen is coated at the base, and the region where antigen is not bound is blocked. Then after that, you will add your sample or you will add antibody. Now this antibody will be linked to enzyme molecule. That means it will be covalently attached to enzyme molecule. And then after that, you will add TMB in this. Now see here, if this antibody is specific with the antigen, then only it will bind to the antigen. And if not, then this antibody will be washed away. Then in the second case, when antigen and antibody are not specific to each other, in that case, no enzyme will be present in this microtiter plate. So next step will be, you are going to detect the presence of enzyme. That means you need to check that whether this antibody and enzyme conjugate has got bound to the antigen or not. When they will get bound? Only if they are specific. And if they are not specific, there will not be the presence of antibody and enzyme conjugate in the microtiter plate. So to determine to detect the presence of this enzyme in the microtiter plate, now you need to add its substrate. So chromogenic substrate will be added here, and if enzyme is present, then this enzyme will act on the substrate molecule and it will convert it into colored product. Now this color you can visually see in the microtiter plate, and you can measure its intensity also by taking its optical density. So this is the basic of ELISA technique. Now there are different variations of this ELISA technique. Now here the ELISA that we have seen is direct ELISA. Why it is called as direct? Because primary antibody is labeled with the enzyme molecule. Then if primary antibody is not attached to the enzyme and a secondary antibody is added in the same plate, that will detect the presence of primary antibody and secondary antibody will be attached to the enzyme molecule, then that method is called as indirect ELISA. Then third variation is antigen can be sandwiched between the two antibody molecules. That will be sandwich ELISA. And last is the competitive ELISA that we are going to discuss now. So now you know what is the basic principle of this ELISA technique. What you need to remember in this is enzyme linked antibody is used, and then second thing is colored substrate is utilized for chromogenic substrate is utilized for generating colored product. So now aflatoxin determination by this competitive ELISA technique. ELISA methods, as we have discussed just now, are based on the ability of antibody to distinguish between the three-dimensional structure of mycotoxins. Now what we have seen here is antigen is detected by using suitable or specific antibody molecule. But this antibody can be detected against variety of biomolecules like from proteins, toxins, and other metabolites also can be detected specifically if you are having monoclonal antibodies for that. So here now we are addressing antigen as a mycotoxin, and wherever you will see that mycotoxin is mentioned, it will refer to the aflatoxin only, because our practical is aflatoxin determination. Now what are the advantages of this ELISA technique if they are used for quantitative determination? One is they are high throughput assays with low sample volume requirements, because microliter is required for measuring the concentration, and high throughput means simultaneously you can proceed with multiple samples. Then second advantage is less sample cleanup procedures compared to conventional methods. Cleanup procedures means, now see, we are using antibody which is specific against the molecule of our interest. So we don't need to remove much of the impurities present in the sample. Whereas in other techniques, these impurities can interfere with the results. Then next advantage is they are rapid, less time is required, they are simple to perform, they are specific by using a specific antibody, and they are sensitive, that is they can detect lower amount of concentrations also. And then next advantage is they are portable for use in the field also due to the easy instrumentation. So they are most quick methods for the detection of mycotoxins in foods and in feeds. Okay, now we are moving towards the competitive ELISA. So now I will explain you the principle of competitive ELISA, which you are going to correlate with the procedure also. So first thing that is done in this competitive ELISA is basically it can be done for both to measure the concentration of antibodies also and to measure the concentration of antigen also. Now, as we are referring to aflatoxin, so we are going to discuss this method as a method to detect the concentration of antigen. So what is our antigen? It is mycotoxin. The first step that you need to do here is you need to have pure suspension of your antigen. So here you need to have pure suspension of mycotoxin, and this mycotoxin should be conjugated or attached to the enzyme. So first solution will be your sample in which mycotoxin will be present, and second solution will be mycotoxin and enzyme conjugate as given in this figure, and here you can see that they are mixed together. Now in your sample you don't know what is the concentration of mycotoxin present, but you are going to add mycotoxin and enzyme conjugate intentionally, so you will add it in known concentration. तो यहाँ पे आपको क्या पता है, माइकोटॉक्सिन का कॉन्सेंट्रेशन सैंपल में कितना है वो आपको पता नहीं है. What you know is just the concentration of mycotoxin and enzyme conjugate. So you will allow this mixture to incubate. Then next thing that you will do is you will take a microtiter plate, and now in this microtiter plate antibodies which are specific against the mycotoxin will be coated at the base, and the area where antibodies are not present will be blocked. Now to this you will add this mixture of mycotoxin and mycotoxin enzyme conjugate. So M refers to mycotoxin and Me is the conjugate. So when you add this mixture, now see, in both type of solutions mycotoxin is present, so both can get attached to this antibody which is directed against the mycotoxin molecules. So here now there will be competition for the available binding sites between both these molecules, and this is why this technique is called as competitive ELISA. Now see, the number of antibody molecules present in this microtiter plate is going to be constant, but what is going to vary? Concentration of mycotoxin which is present in your sample, that is going to vary only, and also mycotoxin and enzyme conjugate is present in the known and constant concentration. So here now they will compete for the binding site and selectively they will get attached. So mycotoxin and mycotoxin enzyme conjugate both will get attached to the available antigen binding site present on this antibody. Then you need to wash this plate so that these unbound molecules will be removed. So now what you have a microtiter plate on which antibodies are coated, to which the mycotoxin and mycotoxin enzyme conjugate has got bound. Okay. Now next thing what you need to do. Now what is the objective of ELISA? After getting attached or after binding of the antibody molecule to the antigen, what you do is you add substrate to detect presence of enzyme in the microtiter. So next step is to add chromogenic substrate, that is TMB, because here commonly used enzyme is horseradish peroxidase and its specific substrate is TMB. So you will add this chromogenic substrate in this microtiter plate, and if enzyme is present, that is if mycotoxin and enzyme conjugate has got bound to the antibody, then only this substrate will be converted into product. So this can be detected by visualizing the blue color that has got developed in your microtiter plate. Now how you will correlate this with the concentration of aflatoxin present in the food sample? So intensity of the color that develops in the microtiter plate is inversely proportional to the concentration of aflatoxin that is present in the sample. So whenever we perform quantitative determination, what we do first is we prepare a standard graph. Okay, standard graph has optical density on the y-axis and concentration on the x-axis. So when you extrapolate the value of unknown optical density of your unknown solution on this standard graph, you directly get the concentration of solute present in your sample. So similarly, we are going to present in this case also. So how you are going to correlate this now? See, first case is your sample has a lower amount of aflatoxin present. So when you add this in the microtiter plate, the aflatoxin which is present in sample is low. So there is more chance that aflatoxin and enzyme conjugate will get attached to the antibody. तो यहाँ पे क्या हो रहा है, अफ्लाटॉक्सिन का कॉन्सेंट्रेशन जो है सैंपल में वो कम होने की वजह से एंजाइम कॉन्जुगेट जो है वो ज्यादा बाइंड होगा. सो ड्यू टू विच व्हाट विल हैपन, more number of substrate will be converted into product and higher intensity of blue color will be observed. Then second case is there is higher concentration of aflatoxin present in your sample. So now in this case, what will happen? Again, the concentration of mycotoxin and enzyme is constant. So the mycotoxin molecules which are present in your sample will compete to more extent and they will occupy more size. That means less enzyme conjugate will be present in the microtiter plate. So less number of substrate molecules will be converted into product. So the intensity of blue color developed will be less if concentration of aflatoxin is higher. So what we can conclude from this? Lower the concentration of aflatoxin in the sample, higher is the intensity of color developed, and if higher concentration of aflatoxin is present in the sample, then lower is the intensity of color developed in the microtiter plate. So this is about the principle of this technique. Now we will see how it is performed. So requirements for performing this technique is first 96 well plate coated with the antibodies against aflatoxin. Then second is standard solutions of aflatoxin in methanol containing 50, 100, 200, 500, 1000 and 5000 nanograms per dm3. Now why these standard solutions are required? To prepare standard graph. So what you are going to do? First, you will perform this technique by using standard known concentration solutions so that you can plot these values on graph. So it will give you a linear relationship and on that you will extrapolate your unknown value. So for that standard solutions are required. Then next thing that you will need is a buffer for diluting standard solutions. You are going to use buffer to maintain the integrity of the solution and to make it stable. Then next is solution of aflatoxin and peroxidase conjugate. So pure aflatoxin will be attached to enzyme molecule, that is peroxidase here. Then you need buffer for the conjugate. Now why you need this buffer is because each enzyme has optimum pH at which it is going to uh, have maximum activity. So this buffer will have the pH that will be suitable for the activity of enzyme. The next thing is solution of substrate, that is tetramethylbenzidine and H2O2. Why H2O2? Because peroxidase transfer the electrons from H2O2 to the TMB and that results into the color change. So for that you need to know the reaction mechanism. Then you need a buffer for washing. Again, this buffer is also made for maintaining the integrity of the enzyme. And then you will require a reagent for process termination. That means to stop the enzyme activity at the end of this assay. Now this is a protocol. So first thing that you are going to do is you will incubate mycotoxin and mycotoxin enzyme conjugate together. Then you will add this mixture into the microtiter plate which is already coated with the antibodies specific against this aflatoxin. Then you will wash out the unbound molecules from this microtiter plate and then you will add substrate in this plate that will be converted into product and you will stop the reactions. All the reactions you are going to stop at the same point so that incubation period will remain same. Then the intensity of color that you will observe, that will be inversely proportional to the concentration of mycotoxin in the sample. Now, how you will measure this intensity? This intensity can be measured using ELISA reader at the absorbance filter 450 nanometer. Now, 450 nanometer is the lambda max for this aflatoxin. So, these optical densities of the sample can now be compared with these optical densities of standards and interpretive result can be determined. Now, how you will be your observations? So, first thing that you need to do is you need to plot a standard curve. Now, as we have seen, these two relations, that is optical density will give you intensity of the color and the aflatoxin concentration will be on x-axis. So, these two things are inversely proportional. So, your graph is going to have negative slope here. So, this is how your standard graph will be there. Then what you will do is you will take the optical density of your unknown sample and then you will extrapolate it. For example, if optical density is 1, then likewise you are going to extrapolate, and if you extrapolate same point on the x-axis, then you will get the concentration of aflatoxin present in your sample. So then how you will interpret your results? So you will in the result you will give what is the concentration that you have determined. So concentration of aflatoxin in given spoiled groundnuts is, like in our example, it is 15 parts per billion. So similarly you will give your value here and you will conclude as the concentration of aflatoxin was determined in the spoiled groundnuts by competitive ELISA technique. That means your practical was successful. So this is all about the practical. Now, as an assignment, what you need to do is you need to first write short notes on aflatoxin so that it will clear your concepts. Then next thing is you need to revise the principle of ELISA technique again. And then you need to write short note on mechanism of action of HRP on TMB. That means what is the reaction that actually takes place when horseradish peroxidase acts on the TMP and why it generates blue color. To summarize now, what we have seen in this session is, first thing that we have discussed is what are mycotoxins. These are fungal secondary metabolites that is having fungal origin, and we have seen the meaning of secondary metabolites. They are produced by the fungus which are not required for the survival, but they are giving added advantage to this fungus. Then second thing is why there is need for aflatoxin detection, because it is hazardous and we should know whether it is present in food or not, so we can see that whether that food is safe for consumption or not. Then third is how it should be extracted. We have extracted by using solvent extraction method, by using suitable solvent, that is methanol here. Then how qualitative detection is performed by detecting its fluorescence under the UV light. And lastly, we have seen how its concentration can be determined by using competitive ELISA technique. So I hope this session was useful for you. Thank you.